So welcome again to all of you. Some of you I know and some of you I don't know. So I'm Bob Mahley. I'm a senior investigator here at the Gladstone Institutes and also on the UCSF faculty. I've actually been teaching the course in the art of lecturing for more than 20 years. And here at Gladstone, the course that I actually give is about a 22-hour course because that allows the students and the participants to practice a talk and then have the talks critiqued. But I've been asked now to condense that 20 hours into 2 hours, and so I will try to give you at least some of the highlights of the art of lectures. This is a topic that I've been interested in for many, many years. I've tried to learn from others some of the best practices and tried to observe what tends to work and what does not work so well. The purpose of the course, as you will hear, is to share best practices that we've learned over the years with respect to giving an oral presentation. Now it's actually called, if I could have the lights down please, it's actually called the art of lecturing because giving a lecture is an art. It's like other art forms, whether it's music, whether it's dance, whether it's painting. Giving a lecture actually is an art. And any good art form requires practice, and then some more practice. But the second part is to have someone critique your talk, someone that will give you an honest appraisal of what you have presented well and what is not so well presented. You will develop your own style for giving oral presentations. There's probably no right or wrong, but I do believe that there are better and best ways to put forth your information and communication. And so I will present to you now approaches that I and others have tried to put together that would reflect excellence as you give an oral presentation. So as I've already said, the purpose of this course is to consider best practices for effective communication for your research. Now we will consider elements of a successful lecture. We will look at the goals of a scientific presentation. We will look at how you structure your talk, and the talk actually is a story. And then we will discuss with you how you build that story, and we will end with a very important discussion, and that's the graphic representation of the material that you're presenting. And I will suggest to you that visuals are the critical issue. They will make or they usually break a talk. There are actually plenty of seats down front. Anybody that just came in, please come on down front. So you can, come on, please. I said people that came late have to sit on the front 2 rows. So, okay. I'm just kidding you, of course. So this is the outline. This is what we hope to accomplish today. Well, what is the purpose of giving a scientific presentation? And you know this, they're obvious. One is to convey information, but the other and the important one is to convince your audience, to convince them to do that. You must develop the ability to communicate effectively. Now, we don't often think we as scientists have a product to sell, but in fact, we do have a product. And that product is the data, is the insights, is the conclusions that we develop in the research laboratory. And that's what we have to learn to sell, because you want your work to be appreciated and understood. Now, I would suggest for your consideration that success requires two elements, and I would suggest to you that 50% of your success depends, of course, on the excellence of your research data. That's without question. But I also believe the other 50% of success resides with your learning the skills, the ability to present your work. I would suggest to you that you should work as hard on your oral presentations, and of course on your written presentations as well, as you do on your experiments, because I can promise you it will pay off. The world of science, as you already know, is a competitive area, and those that are successful are those that develop the skills required for success, and one of those skills is oral presentations. Well, now let's consider some of the elements of a successful lecture. First of all, we all present to very intelligent audiences. And in fact, if that audience does not understand your presentation, it is in fact your fault. It is your ability to present in a way that anyone can understand. In fact, everyone here today is capable of understanding any lecture that any of you would give if in fact you do it well. That's your responsibility as a presenter, as a scientist, to make your work understandable. Now, second, don't make your lecture a mystery story. Make it easy to follow. In fact, a good scientific presentation is just the opposite of a mystery story. And I'm going to suggest that right up front in your lecture, you tell your audience, you tell them what you are going to tell them early in the talk. And then you tell them, that's the facts, that's the research, that's your data. And then you conclude with, tell them what you told them. Tell them what you're going to tell them, and tell them what you told them. I'll say a lot more about this simple principle, but I think this is very important. Now let me also introduce you to the KISS principle. KISS stands for Keep It Simple. That is a key to a good lecture. Some people say that that additional S stands for stupid. Keep it simple, stupid. And if you remember nothing else from the talk, remember the KISS principle. Listening to a lecture is hard work, so help your audience. Don't load your talk with a bunch of extraneous facts. You're not here to prove that you know everything about a topic. You are here to present the material that I need as the audience, that I need to understand your lecture. And fourth, you're going to create a story, and we'll talk about how to structure a story. Manuscripts are where you document details. That's where you give 0.025 molar tris, pH 7.25. That's not what you give in a talk. Those are the details. They go in a manuscript. Most of the time, those details are not so important. In a talk, tell your audience, as I've already said, tell them only what they need to know in order to understand your research. Almost never use a figure or never use a table from a manuscript. They're almost always too complicated, too difficult to explain. They're loaded with too much detail. Well, let me share with you now how I prepare a lecture. As I've said, you'll develop your own style. But what I do is I determine the message of the talk that I want to give. And what do I mean by the message? That is what I want you to learn. That's what I want you to understand, and most importantly, it's what I want you to remember from the story that I build. And we will, in fact, build a story around the research. And then I collect all of the data, all of the facts. What you need to know goes in the background. That sets the stage for the presentation of your research. I actually prepare a series of slides. They may simply be a piece of paper on which I've written, show the data from what such and such. Maybe I haven't even designed the slide yet. But I put together all of these slides, potential slides, in a stack, and then I can shuffle them. You almost always put together too much material for the time you've been allotted, whether it's a 20-minute talk, a 40-minute talk, or whatever it is. So if you have these slides, these potential slides, you can shuffle them, you can add, you can subtract. That actually serves for me as a way to organize my talk. It actually serves as an outline, and in the process, I'm developing the visual aids that are required to tell the story. And fourth, I always write out my talks. I have written out every talk that I have ever given. I won't tell you how many years ago I started doing this, but it's more than a few. But I've written out every talk that I've given. I would suggest that most people don't do this, I understand that, but I would suggest that at least when you're starting out, that you write out your talks. Now, that does not mean you read them. In fact, that does not necessarily mean you even use all of those notes, but I always have my notes. You can't really see them, but this is a page of my notebook with this slide and with the points that I want to make to the class. Now, why do I do that? Well, one, it helps me to organize my thoughts much better than I can in just thinking. Maybe that's my problem. But I feel that I organize what I want to get across. I choose the words better, I hope. But the most important reason why I do this is because I'm a very anxious presenter. I get very nervous when I present. I have on occasion forgotten where I was in a talk. All I have to do, not to read my notes, but to refer to my notes. So I would suggest to you, at least on occasion, when you're getting started, try this. Well, let me repeat for you then. The goals of a scientific presentation are to send a clear message. I've already said it. That's what I want you to learn. But importantly, that's what I want you to remember. One of the biggest compliments that you can receive as a presenter of your data is to have someone to leave the auditorium and to be able to say, I learned X, Y, and Z from that lecture. So that's my responsibility to make sure you know what I want you to learn. That's the message. And we'll talk a lot more about the message in a few minutes. I'm convinced that if a person can walk out of this room and say, I learned X, Y, and Z, the next thing they're going to say or think is, I'm glad I took the time to attend the lecture. And the second point is, the goal is to present a clear story. It is a story, and it must go logically in its organization. The story explains the message. The story validates the message. The story makes the message clear. It must flow logically like any good story. Well, what do I mean by the message? And here again, this is another key point to remember. The message is the main point of your talk. As I've already said, it's what I want you especially to remember about my research. The message should be stated on a slide early in the talk. I'm going to give you parts of a demonstration lecture as we go through this exercise. But the message slide that I will share with you in just a couple of minutes will appear on about the 6th or the 7th slide in this demonstration talk. It will be a sentence. I will have it strung together in a way that I could write it out and I will show you. I'm going to repeat it at least 2 times, but most of the time 3 times. In fact, in this talk, this demonstration talk, part of the talk, I'm actually going to show my message slide 5 times. And this will help you to get the message. As I've said, your talk is not a mystery story. Your talk is one in which right up front I'm going to tell you what I am going to tell you. That's in the introduction. That's the message. Now, a lot of people have told me after I've said this and other occasions, they say, but if I tell the audience what I'm going to tell them, they won't be interested. They'll be bored. No, I swear to you that's not the case. What it will do is, it's just the opposite. I now know if you tell me what I'm going to tell you, I now know what to focus on. I know I can follow the talk more easily. I can relax. I can pay attention to the data. I can actually be critical of the data. I know what you told me you were going to tell me. Now, in fact, did you tell me what you told me you were going to tell me? So I can relax. I know what you're going to tell me or what you hope that I believe. Okay, so let's focus on putting the story together. The story has 3 parts: a beginning, a middle, and the end. This is a famous formula. It sounds simple. It is simple. There is an organization that started a long, long time ago. It's called the Toastmasters Club. I don't know whether any of you have heard about it. There are other organizations like this now, but they have taught essentially every CEO and president of all the Forbes 500 companies around the country. They've taught them this famous formula. That is, there is a beginning. And what is the most important part of the beginning? I've already said it several times. Tell your audience, tell them what you're going to tell them. Then the middle part, that's the facts, that's the data, that's your research. That's what you want to get to, that's the fun part. And so you're going to tell them. And then at the end, you're going to tell them what you told them. So, what's in the beginning? Remember that the beginning is a step-by-step build-up to the message. Most of the time, you actually can't give the message of the talk in the first slide or the second slide. Most of the time, the audience needs some background information in order to understand your message. And so in the beginning, before you give the message, you need to have an introduction. You need to state the general topic, and then based upon this general topic, you're going to build the background. That may take 4, 5, 6, 7 slides. It depends upon the complexity of the subject and the length of the talk. But the background has all the specific facts and all the definitions that I need to know. I need to be convinced that it's important that I understand what we are talking about, and then I can present the message. As I said, you can't give the message usually in the first slide. And in the talk, demonstration, parts of the demonstration talk that I will give you, it's in the 6th or the 7th slide. Now, I can't actually give you a full 40-minute lecture on top of the instruction that we're talking about now. So I'm actually going to pull out parts of a talk that I gave 6 or 8 weeks ago, and I will illustrate for you components in the beginning, components in the middle, and components at the end, and I hope that's helpful. So what is the general topic of the lecture that I'm going to give? Well, the general topic is apolipoprotein E4 and Alzheimer's disease, mitochondrial dysfunction, and altered CNS metabolism. The title is informative. I don't actually believe in clever, funny titles. I'm too conservative. I want you to put a title on the talk that actually is informative, that tells me what we are going to talk about. So I'm going to tell you, obviously, from the title, about a protein called apolipoprotein E, or ApoE. Now, many of you, probably most of you, have never heard of this protein. That doesn't make any difference. I don't care, that's okay, because I'm going to tell you what you need to know about this protein before I present my data. And I will give you that information in the background. From the title, you also know that ApoE is related to the disease called Alzheimer's disease. Many of you, almost all of us, know something about Alzheimer's disease, but I'm going to describe it in a little more detail to put it in the context of what I want to talk about. Alzheimer's disease is a terrible disease. It's increasing in prevalence because of an aging population worldwide. And so I will tell you something in the background about Alzheimer's disease. And from the title, you know that this protein involved in this disease affects mitochondria and other aspects of metabolism. So let's talk more now about the beginning. Before I go into the details about the beginning, let me tell you about what I call throwaway comments. That's the first 15, 20, 30 seconds of your talk after you've been introduced. Good afternoon. My name is Bob Mahley, and I am an investigator at the Gladstone Institutes and UCSF. It's always a pleasure for me to speak to the Department of Neurology at UCLA. Why are those 2 sentences, 20 seconds, maybe it was shorter than that, why are they important? Well, they allow the audience to settle down. They allow the audience to start focusing on me and what I want to talk about. You just ran in from the laboratory. Your experiment wasn't going so well, or you're wondering when you can get home to watch the game, whatever it is. You got all sorts of things running around in your mind when you come in here and sit down, but I want you to listen to my lecture. And so I break the ice. So what do? I allow you to hear my voice. I allow you to settle down. And I also, in the second sentence, I acknowledge you as the audience. I'm glad to be here, and I'm glad to speak to you. Other possible components of the introduction, you can expand on who you are. As an investigator, I've studied this protein for years. Why it's important? Alzheimer's is a terrible disease, fastest growing neurodegenerative disorder facing humankind. Why should you be interested? Because there's no treatment. And it's doubling, the aging is doubling, tripling, quadrupling in prevalence because of an aging population. So, we're still in the beginning. I've given the 15 seconds of throwaway comments to allow you to settle down and to focus on the lecture. You've been introduced to the topic, and now I must provide you with the background leading to the message. What do you need to know to understand what I want you to learn, what I want you to remember? That is what I've said here. What does the audience need to know to understand my story? So up front in the introduction, we're talking about the second, third, fourth, fifth, sixth, seventh slide, something early. You're going to define all the terms. You're going to set the stage for the research that I want to present, or that, if you're the speaker, you want to present. And remember, you're leading up to the message. Now remember, what's common knowledge to you with respect to terms and so forth may not be common knowledge to everybody in the audience. We often make a mistake by assuming everybody is expert in the topic like we are. But in fact, we almost always speak to general audiences. There are people in the audience that come to hear you that come because they want to learn. The title is intriguing. It's an interesting topic. They're not expert. And so you must help them by defining the terms and setting the stage. Don't use lab jargon. This is not a lab meeting in most cases. This is much more serious than that. And I also suggest not to use what I call alphabet soup. We use that all the time. What is chip-seek? It took me forever to figure out what that was, going to all these lectures, because that's all the speaker said. Well, now this is the chip-seek data. Well, what the heck was chip-seek? Well, I now know. But when I first started hearing, I didn't have a clue what it was, so I saw the data. It meant nothing to me. PKPD, that's a term that's used. We all use that. Maybe you've heard it, maybe you don't. But it's my job to define it for you. Don't use a term that you don't define. And define it in writing on the slide as a footnote. So, forget jargon, forget alphabet soup. So now we're back to the general topic. ApoE4, Alzheimer's disease, mitochondria, CNS metabolism. So what do you have to know to understand my research? Well, you've got to know about this protein. I realize most of you have never heard of it. That's fine. I can't start telling you what it does if you don't know what it is. So I'm going to explain that to you. You've got to know something about Alzheimer's disease and how ApoE fits into that. And you need to know a little bit about mitochondria and metabolism. I must tell you the facts then. I must tell you that ApoE is a protein in which there are 3 isoforms. The title included ApoE4, so you could have guessed there was probably a 3, and in fact there is a 2. Don't ask me why there's no 1, but there's no 1. So there are 3 isoforms. And I've got to tell you about the structural differences among these isoforms that determine the function and the relationship to Alzheimer's disease. I must put in the background, before I give you my message, I must tell you something about Alzheimer's disease, a neurodegenerative disorder associated with dementia. But importantly, this is an important protein because 65 to 80% of all patients with Alzheimer's disease have the ApoE4 form, and this represents a new therapeutic target. So let me show you what I put in the background of my demonstration lecture. I can't give it all to you, but I can give you parts. This is the second or third slide. It's in the background. There are 3 isoforms of this protein, E2, E3, and E4. ApoE3 is the most common form in humans, but ApoE4 is not rare. It occurs in about 25% of individuals. These forms differ by single amino acid interchanges that occur at residue 112 or 158. And especially important for our discussion today is that ApoE4 differs from E3 by having an arginine at residue 112 instead of the normally occurring cysteine. And this is associated with the development of Alzheimer's disease. And I will show you that this single amino acid interchange causes ApoE4 to become neuropathologic. And ApoE4 is actually the major genetic risk factor for Alzheimer's disease. Okay, so now you know everything you need to know about ApoE4 to understand my research. It didn't take long, but it was critical, or you wouldn't have understood why I was doing what I'm going to show you. So what is Alzheimer's disease? I've already told you some of this. Most common, devastating, dementia, loss of CNS neurons, prevalence increasing, no effective treatment. 5 drugs on the market, none of them, in fact, have a significant impact on the course of the disease. This is an important topic. That's why I said that. Okay, a little bit more about Alzheimer's disease. There are multiple causes. It's a complex disease. There are multiple causes. There are several pathways that are considered to be important. I won't go into detail here, but I'll mention amyloid pathway. I'll mention the neurofibrillary tangle pathway. But what is the focus of the talk today is the relationship of ApoE4, because we have shown that it has a direct toxic effect in causing neuronal cell death. I will also show, now we're at about the 5th slide in the presentation, the impact of Alzheimer's that it has on the brain. Shriveled, a loss of volume, actually a loss of nerve cells and fiber tracts. I will explain this slide to you. I won't do it right now, except what I just said. This is the background leading to the message. So now I'm going to tell you, I'm going to tell them, the audience, what you are going to tell them. I'm going to tell you what I'm going to tell you. Okay. I've step by step built up to the message. The message is stated up front. In this case, it's the 6th or 7th slide. It's going to be shown on a slide. I'm going to use it several times. In fact, I'm going to use it, I think it was 6 times in this lecture. And it's going to allow you to easily follow the talk if the message is well stated, well focused. You can now focus your attention on how good is my data without worrying about the fact you didn't understand what I was talking about. So what is the message of this demonstration talk? Now, I've written it out in a sentence. You wouldn't show it this way on a slide, but it's in a sentence because this is what I determined when I was asked to give this lecture a couple of months ago, 40-minute lecture on a topic. I wrote out this message. ApoE4, first of all, assumes an abnormal structure. Second, that abnormal structure causes the ApoE4 to undergo neuron-specific proteolysis, generating a series of neurotoxic fragments that cause mitochondrial dysfunction and cytoskeletal alterations, leading to pathology. This is a complex sentence, I understand that. And as I said, I would never show this on a slide. But this is what I wanted you to learn. I'll present this in a much better form in just a minute, I believe a better form. That's what I wanted to say, for you to say when you leave the room about the talk. Now it's my job, in the middle of the talk, to prove these points. That's what I'm going to prove. I'm going to show you my data that has been generated here at the Gladstone to prove these points. But I'm going to convert this message slide into a much more friendly slide, a much more usable form, and a form that I will use 5 or 6 times during the talk. It's exactly the same thing. ApoE4 assumes an abnormal structure that causes it to undergo neuron-specific proteolysis, which generates a series of neurotoxic fragments leading to mitochondrial dysfunction, cytoskeletal alterations, and neuropathology. It's still a complicated message, but you'll get it because you're going to see this slide 5 more times. But I'm not going to bore you. This won't be boring, I hope, that you will then be able to understand the message. So one of the versions of the slide that I'll show when I've gotten to the point of discussing the data on mitochondria, I'll show the slide again. I will highlight that part in yellow. It's my outline. It leads you, it helps you to follow where I am. You build the story then by each step-by-step point through your research data. Now we're to the major part of the talk. We're to the middle of the talk, the tellem. As I said, this is where you wanted to go right away. I made you slow down a little bit and show 7 slides before you got to the tellem part. But now they'll be able, I hope, to get the message. I've spent a lot of time on just telling you about the beginning. And the reason is because I believe that most talks fail because of a poor beginning. The audience doesn't understand where you're going and doesn't understand the background, have enough information. Now, how much time do you spend on each of these 3 components? Well, let's say you've been asked to give a 30-minute talk. So a 30-minute talk, commonly, depending on the complexity of the subject, the beginning will take 5, 6, 7 minutes. And it will encompass 5, 6, 7, 8 slides. The middle, that's the big part. That's going to take you 15 to 20 minutes of the 30, and it'll be 15, 20, a few more slides than that. And then the end, again, how complex you make the conclusions and the summary, it will take another 5, 6, or 7 slides, 5, 6, or 7 minutes. So that's roughly how you divide the talk up. So let's remember now, we're telling a story, and we're at the middle. This is the fun part because we're going to tell them our research results. So you describe the experiments, and then you must link the experiments. You don't, as you know, I'm not telling you anything profound. You know, you just don't show one experiment and then go to another experiment and so forth. You have to link them. This is a story. You have to, these are chapters in your book. You have to build it that way. Now, you don't have to present your results in the order you did the experiments. I assume you understand that. Because the 4th set of studies you did may be the most important. The 2nd and the 3rd, least important. You may show them, but you may show them 2nd or 3rd. It's a true story, that's for sure, but it doesn't have to necessarily be in the order that you did the work. Each experimental set of data needs to be introduced, each experiment, if you will. And we follow, as you know, the scientific method. And so there is a question or a hypothesis that's stated. Then we show the experiment, maybe this is the methods, and then the results, and then we give the answer to the question. But I would like to suggest a twist on that format. I would like to suggest, just as we've already talked about, that once you state the question, you give the answer to that question right then. You tell them what you're going to tell them. And then you tell them, this is about each experiment. And then the answer is you tell them what you told them you were going to tell them. This just helps your audience. It helps to understand. It takes this mystery out of it. It makes the audience relax. Do try it. Try to do that. So what would be an example for my talk? The question is, how does that single amino acid interchange in ApoE4 cause neurotoxicity? That's the question I would state before I show you the data, and I'm going to give you the answer. As I will show you now, this change causes it to undergo neuron-specific proteolysis and the generation of toxic fragments. And then I show you the experiments. Did I prove it? Did I do what I said I was going to do? Well, you sit there and you listen and you see if, in fact, I did it or not. The middle is where you support, defend, and make specific your message. So the second part, the tell 'em part. You have to link those experiments one to the other, and you link back to the message, the broad message of your presentation. It is a story, and you're developing a storyline, and it must flow. Well, how can you link one set of experiments to another? Sometimes it's a spoken link. It can be on a transition slide, but it can be spoken. So I've just shown you data related to our findings at the level of the tissue. Now let me go to the molecular. Or I've shown you in vitro data. Now let's look at the in vivo studies that do thus and so. Or, so to go back a little further, then let me tell you, or now the next step in the logical progression was to so forth. But what I like to do is to repeat an outline slide. In fact, I like to convert my outline into my message. And that's what I've done, and I've already shown you that. For example, I would use this slide. So I've already described for you the significance of the abnormal structure of ApoE4. Now let me present to you how that abnormal structure leads to neuron-specific proteolysis, the generation of neurotoxic fragments, and the effect on mitochondria. So I would use that slide again and again, as I said, 6 times. So in summary then, of the middle, the tell 'em part, this is where you're going to describe your experiments. This is the meat of the talk. I'm going to tell you what question I'm going to answer. I'm going to give you the answer. I'm going to show you the results, and the answer will be what I told you I was going to tell you. And then I'm going to link the experiments, one to each other, and I'm going to link back to the message. Well, how do I prepare the middle of the talk? It's just like I've told you for the general outline of the talk. I use my message slide as a guide to develop the story. I collect the data in the form of slides, raw data, maybe just a sentence on a piece of paper as to what I want that data, what component of the data I want to show. I spread out all of that data. I always have to eliminate some. You never have enough time to present everything you would like. And you actually begin at this point to design the slides, and you develop the logical flow of the story. So let me go back to the demonstration lecture, at least a small portion of that lecture, illustrate using the message slide to introduce a new topic. And this is actually the first set of data. I've just entered now the middle of my talk. What I will do now is to compare the structure of apolipoprotein E3 and ApoE4 and describe for you the abnormal structure of ApoE4 and discuss how it ultimately will lead to neuropathology and Alzheimer's disease. So that's how I would introduce this first set of data. Now, as I've already said, ApoE3 and ApoE4 differ by single amino acid interchanges that occur at residue 112, where ApoE4 has arginine at this site, ApoE3 has cysteine at that site. And this single amino acid change has a profound effect on both the structure and the function of ApoE4 as illustrated here. During the actual lecture, I would present to you how that structure differs, because in ApoE4, because of this arginine, the amino terminal domain and the carboxy terminal domain actually interact with each other, causing the abnormal structure. Actually, that's what stimulates the proteolysis and the generation of the neurotoxic fragments. So I would explain all of that to you. I would explain the nature of that ionic interaction. But that doesn't occur in ApoE3 because of cysteine at 112. Obviously, as I said, I cannot go into the detail of what I would do, but I've tried to explain that to you. But also in this section, I would present data that we have small molecule structure correctors that can insert within the structure of ApoE4 and disrupt this amino terminal, carboxy terminal domain interaction, converting ApoE4 to ApoE3-like molecule, and in so doing, reverse the detrimental effects of ApoE4. And I would show you the data supporting that set of statements that I just made. I would go back then to the message slide, and I would say something like, now that I've shown you the importance of the abnormal structure of ApoE4, let me demonstrate how this structure leads to neuron-specific proteolysis and the generation of the neurotoxic fragments. And then I would present the data that support that. So that's the beginning we've talked about. We've talked about the middle, now the end. The end of the talk is tell them what you told them. And it's the same message. It is the message that you told them on the 7th slide. I am now on the 42nd slide of this demonstration lecture, and I'm telling you in the same words the message that I have repeated, and I can go further and talk about implications and so forth. So this, in fact, would be my last slide. I have described for you the importance of the abnormal structure of ApoE4, in which there is domain interaction that leads to the abnormality that stimulates the neuron-specific protease. The protease, as I have shown you, is a serine chymotrypsin-like protease. The fragments that are generated by this fragments range in molecular weight from 12 to 29 kD, and they are lacking the carboxy terminal domain. Those are things I would have told you in the lecture. And they impact the cytoskeleton and the mitochondria, leading to neuropathology and Alzheimer's disease. So we're at the conclusion. We're at the end. You've told them what you said you would tell them. Now, you can end with interesting points that you want to make. It depends on your lecture. What's your interpretation of the data? Is there a broader implication for what you've learned than was obvious? How does the science contribute, or how does your result contribute to biomedical science? I could talk more about small molecule structure correctors that are being developed as a therapeutic. And also, Have you proven the hypothesis? Have you answered the question? Sometimes you don't, and this is the time to talk about them. My data does support X, but you know there are still questions about Y, and I must do more. That's a very interesting way to end and stimulates a lot of discussion. So the structure of your talk, it is a story. It has a beginning. You build up slowly in the talk, allowing your audience to come with you. You give the introduction and you give the background. Everything they needed to know to understand my data. And then you give the message. Tell them what you are going to tell them. And then to the middle of the talk, you describe each experiment. And along the way, you give the answer. At the same time, you give the question that you are going to show them you answered. And then you end, you show the same message slide. Use the same words. Make it easy for the audience. As I've said, it's hard work coming and listening to talks. Help me to understand, and you will profit greatly from that. So in the end, tell them what you told them, and so forth. Ask yourself when you get ready to prepare a lecture, what do you want your audience to know? What do you want your audience to learn? What do you want them to understand? And fourth, and most importantly, what do you want them to remember? And the other point, as I stop and ask for questions now, is remember the KISS principle. Keep it simple, because it's hard work to get your message. Don't make it a mystery story. Make it a fun story that flows. So I will stop there before we go on to discuss graphics and preparing your slides and visuals, to give you an opportunity to ask any questions that you might have at this point. Are there any questions? Yes, please.
Very often these talks just for 10 minutes. 10 minutes you have to... So how would you approach those?
Yeah.
Is that in the end, it's great, would you make a small version of this, or you kick things out, especially this repetition, where you would lose time?
Yeah, a 10-minute talk, which is, of course, common for major meetings, a 10-minute talk is actually the most difficult talk, because you have 10 to 12 to 13 slides. That's all you got. If you start loading up 15, 20, you're dead. The other point about a 10-minute presentation, always write it out, because every 10 seconds counts. You should have thought out the actual wording. Now, you will not be able to cover a complex topic like I just gave you a demonstration. That was actually a 40-minute lecture and 20 minutes of discussion. So a 10-minute talk, you will focus on only 1 or 2 points. You may have to summarize a lot, not glossing over, not giving enough information. But abstracts, talks, 10-minute talks are actually the most difficult, and you have to work very hard. But there again, that is really important to give the message. It has to be a simple message, because you can't cover 6 different areas, or however many areas I actually covered, at least 6, 1, 2, 3, 4, 5, 6, yeah, 6. 6 topics I covered and showed experimental data in that lecture. You can't do that. So you will have to simplify it and make it. But do give a message. Now the message slide, I said my message slide was too complicated to just be written out the way I did it to just introduce it to you. But you could convert a slide like that into ApoE4, bullet, abnormal structure, bullet, enhanced proteolysis, bullet, and that would have simplified it, and you can do that. So, but do develop a message and give it in a 10-minute abstract talk. It's your third or fourth slide, at least. And your second slide after your topic slide is the background you need. You may only be able to give 1 or 2 slides of background, but almost always you have to have some background. Okay, is that helpful? Okay. Other questions? Yes, please.
What if you have your research is still in progress and you don't quite have a clear message yet, and you don't know what you're doing?
Yeah, well, those are research in progress presentations, RIPS presentations. They're the most fun and they're the most valuable for you because you don't have everything cut and dried. My talk was pretty, the facts were pretty strong, represents 20 years of work, so it's, it's pretty strong. Where most of you are, where you're just developing your career and you've just done a few experiments, that's what you present. And you just up front say, I don't have a complete story. I will show you where I am now in my research, and I will look forward to the discussion because that will help me as I shape the next experiments. You don't have to have complete stories. That is a mistake for a young investigator to always believe. You will learn a lot more by simply being honest, showing what you have, and hopefully the questions will lead you to the next set of studies. Yes, in the back.
Suggest developing one major story in line with your presentation, or how would you approach that?
Yeah, job interview talks. It's a hard question to answer because I need to know in order to answer the question, what was the job description that you applied for? Now, if it is for a neurophysiologist, say, or a neuroelectrophysiologist, and obviously I wouldn't be being considered if I didn't have something related to electrophysiology. And so the job is interested in that. I would pick from my research portfolio that which fits the job description. And then you can always say, in addition, I have done thus and so. But I will not go into it. In fact, on your second slide, you could, in fact, say you've worked on 3 different areas, and they're not totally related. As you said, you're exploring the world of a particular topic. But and so you could list all 3 of those, just so they know in case a search committee member is actually interested, not in electrophysiology, but in sequence homology or whatever. So they would know that, and then they could ask you about that. Yes, job talks have to be focused. They need to be as good a story as you can make. It doesn't have to answer everything. You just have to be honest. That's the important point in giving job talks, is be honest. What you know and what I don't know. There's lots of things we all don't know. There's nothing wrong with that. So one other point to make on this lecture that your question brings up. Consider every talk a job talk. Every time you give a talk, you consider it you're interviewing for a job, because you don't know who's in the audience. We all go out and try to look for you bright young people. And what are we looking for? We're looking for a good story and a good person. So you don't know whether when I'm come to hear your lecture, whether I'm looking to recruit you to come to the Gladstone as a faculty member or whatever. So consider every talk that you give is a job talk. That means you have to work really hard on every talk. There are no sloppy talks. Okay, anything else before we, yes, please.
Say you have two projects, maybe on different diseases or something. Would you consider them as two different stories in one talk, or?
They ought to link in some way. And maybe they would. Maybe it's neurodegenerative disease, it just happens to be two diseases. So as long as there is some theme that links things, I think you can present two stories. But they do need almost, now what I'm saying is not 100%, of course, because I don't know the specifics. But if you can link them, link them. If you can't, just say, I'm going to present this one. Because you do want to, in a job talk, you do want to know to give enough detail so they know that you understand what you're doing. So, and of course, it depends on the length of the talk. Some job talks are 20 minutes, some are 30, some are 40. And you don't know until you're told what it is. Okay, well, let's talk now about slides, graphics. Oops, let's see, what did I do here? Here we go. And this is an important statement that I put here. Visuals will make or break a talk. The quality of your slides. The quality of your slides reflects in a lot of people's minds, and especially mine, the quality of your work. Slides that are poorly prepared, they're loaded with a bunch of stuff that I don't need to know. They're difficult to read because the font size was too small. They detract from your excellence as a scientist. Poorly constructed slides suggest to me and to many people sloppy science. I consider that the preparation and the use of slides to be one of the most important considerations for you as you put together your presentation. So I want us to think about the best ways to present data graphically to communicate your story. Now, slides do not replace the words. They do not replace the discussion, but they are used to strengthen and to highlight the results that you're presenting. So let's consider some of the general guidelines. Excuse me just a second. Some of the general guidelines. The KISS principle, again, keep it simple. Don't put things on slides that I don't need to know about, and don't put things on slides that you don't describe. Because I'm going to try to read everything on the slide. That's just human nature. You try to look at everything. But if there's a whole part of the slide that you really aren't going to tell me about, it just detracts from the message that you wanted to get across. Models are extremely helpful. If you can draw a picture of something, draw a picture. And it's really true. It's trite, and we all hear it. Picture's worth 1,000 words. It certainly is in an oral presentation. Large enough to read. There's nothing that irritates many audiences, including me, than to have someone describe a slide and I can't read a thing on the slide because the font size is all too small. It is so irritating. So you have to consider the size of the room, the size of the audience. Now, I'm going to suggest a way that you don't have to consider the size of the room or the size of the audience. That is, you choose a font size that's big enough to be read in this auditorium or in a conference room. You can do it. You don't even have to think about whether it's a huge auditorium or not. You choose font sizes, 36, 32, 28, 24, nothing less. And it can be seen in every size, almost every size of a room. Use a slide appropriately. When you practice your talk, practice with a pointer. What are you going to point to? When you use a slide appropriately, you lead the audience through the slide. And don't circle. I think I'm borderline epileptic, but people that circle drive me nuts. I actually quit looking at the slide. Now, what do I mean? Make sure it's large enough to read. Got it? No. Just lead me large enough to read. Now, why do people circle? Because they're nervous, just like me. I can't hold my hand out here and not jiggle. But I didn't jiggle when I pointed that out to you. How did I do that? I had my arm by my side. I stabilized the pointer. I'll use a podium if it's appropriate to put my hand on. I can't do it this way. I really do. I'm not doing that for illustration. I cannot do it, and most people can't. Every presenter is anxious or nervous in some way, and don't worry about it. It's actually good, because you'll work harder if you're a little nervous. When you get too comfortable, you do a lousy job. So don't get too comfortable. Okay, where am I? Okay, more guidelines. Don't speak a long time with the wrong visual on the screen. Use a transition slide, or use, a simple word slide or something. Everybody's mind wanders. Don't give me a chance to drift off back to the lab, to the experiment that wasn't working so well. Don't give me an opportunity. Keep me moving with you. Keep the story flowing. Don't let me get distracted. Transition slides, they're good for breaks. Questions are great. Typically, use each slide, should be on the screen. 60 seconds, that's how you can come up with the number of slides you need for the number of minutes that you talk. 30-minute talk, 30 slides, 32, 33, 34, that's the range, because you'll average something like a minute on each slide. Now, some slides, of course, where it's simple words or questions that you're stating, they're 15, 20 seconds. The other thing about showing slides is don't flash the slides too quickly. A lot of people, I'm one, take notes during most lectures, and I'm sometimes looking down. And if someone's going so fast through slides, by the time I look up, I just realized I missed a slide. And that, of course, must have been the most important slide of the talk, and I didn't even see it. So don't have any of these 4, 5, 6 second slides, because, for one thing, it's too fast to focus. Sloppy slides, I'll say again, suggest sloppy laboratory procedures. The lecture room. When possible, be comfortable with the lecture room. If you're giving a lecture here at UCSF, you almost always, or at Gladstone, you almost always know the conference room that you're going to speak in. Make sure you visit that conference room. Make sure you know where the lights are, how everything works that is necessary to allow you to be efficient in setting up and presenting. Whenever I'm a visiting professor, I'm usually shown around by a postdoctoral fellow or sometimes a student that ushers me to where I'm supposed to go. Early on in that visit, in fact, almost immediately, I asked the person, would it be possible to go by the lecture hall so I can just see what the setup is a few hours in advance, maybe, of giving my talk? There's nothing wrong with that. It's usually very easy to do. And so I know what the setup is. What sort of a microphone is there? Do I have to be worried about the projection of my voice? Sometimes I want to know where the light switch is, because if I've got a bunch of, say, electron micrographs or something, I may actually want to switch the lights off or switch them lower. Sometimes it's not convenient. Sometimes you just can't do it. Oftentimes, IT people will set you up and then they leave. And so you're sort of stuck in the middle if you don't know how things work. Practice with the equipment. Always practice, if you can, with a knowledgeable colleague. Practice with a clock so you know how long your presentation takes. I will suggest that a 20-minute talk will probably take you 10 hours to prepare. Plan for that. Now, I don't mean sitting at your desk for 10 straight hours, but over a period of time, because talks often evolve in your thinking. Slides often evolve. Slides sometimes take a longer period of time to make. So think about that and plan the time so you have enough time in order to really have a well-prepared presentation. I think one of the most important things that I can tell you in this section of our discussion is to practice, and then practice, and then practice, and then practice with a knowledgeable colleague that will honestly tell you what's good and what's not good about your presentation. Absolutely do that before you do a job talk. And make sure that your slides project well. Some more general points. Now, general points about the design of slides. Simple text, keep it simple. Bullets, not sentences. Readable size, fonts, very important. Use a slide just like we read, from top to bottom, from left to right. Don't use tables from a manuscript. They almost never work. The title of a slide can be of two different types. It can describe the slide or a method, or it can give a conclusion. This is very important. The title of the slide is the first thing that you see when you see a slide, because it's usually, almost always, in larger font size. Sometimes it's actually highlighted in color. We'll talk about that in a minute. So the title of the slide is very important. As I said, there are two types of titles. A description of the data that's being presented on the slide. That's certainly reasonable in some cases. I actually prefer that the title be an interpretive statement of the results that are being shown on the slide. And why is that? Because sometimes I look at data that someone's presenting, and I honestly do not understand the methodology. I know I don't know all methods that are used. And so sometimes I can't actually interpret the data for myself. Now, hopefully the presenter will have led me through the data well enough so that I will understand it. But if he or she doesn't, then I rely on the title. The title tells me, an interpretive statement, tells me what the presenter wanted me to learn from that data. Maybe I can't get it, but at least I get what he or she wanted me to know. So here are a couple of examples, or several examples of slides. They almost all have something to do with ApoE. because those are the easiest, those are my slides, so I can pull them out and show them. Here's a slide. It's actually not important what the data is, but this is a slide that's descriptive of the method. It's an ApoE immunoblot of the supernatant fraction of the human Alzheimer's brain. What it shows are those fragments that I told you about that I didn't show you, and their characterization. They lack a certain part of the molecule. Now, if you're not used to looking at immunoblots, and if I don't do a good job in explaining this slide, then the next most important thing is you may have to rely upon the title. And the title is Carboxy-terminal truncated forms of ApoE are generated in the Alzheimer's brain. So I got it, even though I really am not expert enough to criticize the data, and maybe I didn't explain it well enough. Now, here's another descriptive title. This is mice put in a water maze, and they swim around. They have to find a hidden platform, and they have to find the platform in a number of seconds. You compare how well they learn by how long it takes them to find the platform. And so what you're looking at here is the performance of 6-month-old female ApoE3 and ApoE4 mice on day 1, 4 different trials in a water maze. And the E4 mice don't learn so well. That's the point. Now, it would have been easier. Suppose you don't understand the water maze. You've never seen it. You don't, and I don't do a good job of explaining it. Then the thing you needed to know was ApoE4 female mice perform poorly on the water maze test. So the title can be very helpful to your audience. Keep it simple. All right, other types of slides. Model slides. A working model, we've already said. If you can draw it, so much the better. A picture's worth 1,000 words. Use models wherever possible. Now, for example, I have various versions of the slide that I showed you to describe the structural differences between ApoE3 and ApoE4. Some are more complicated than others. This is a very complicated slide. I used this at a Keystone meeting in which they really wanted to understand the nature of this ionic interaction in ApoE4. I didn't present that detail in the demonstration lecture that I gave as part of what I was talking to you about because it wasn't critical. It just was a complication that you didn't need to be involved with. Now I can even use a simpler slide, and I do use a slide like this. Same thing. This ionic interaction between arginine 61, glutamic acid 255 in ApoE4. It doesn't occur in ApoE3. So you can create different versions, different degrees of complexity that you want to show. Now this is a slide that a student in my Gladstone course used in a demonstration lecture that they gave. It was a study about HIV infection in rats. This was the method slide. It's an absolutely beautiful slide, in my opinion, because it told me everything I needed to know in about 40 seconds. So, what did he say? In the anesthetized rat, I injected either intravenously or interperitoneally a dose of the HIV virus, followed its course through the blood, and localized it in the spleen, the thymus, and the brain. 35 seconds. Everything I needed to know. Beautiful slide. I know everything I need to know. Okay, here's a slide that I consider not so beautiful. Okay, this slide was used also in the course. It has to do with NF-kappa B, complex subject. It's a great subject, but it's a complex subject. This presenter had 20 minutes. Couldn't possibly lead me through the complexity of NF-kappa B. All she wanted to tell me about was TNF alpha driving gene expression. But she showed the class all of this stuff. Now, it's all true, and for a Keystone meeting, yes, you show that. For a 20-minute general audience presentation, you can't. It's too much detail. You put the receptor that you want to talk about, and then you simply list other or something along those lines. You don't make it complex. She also didn't need to tell me about this IKK signalosome. Very complicated. She didn't explain it. She just sort of flashed it. And so what I'm doing is I'm looking at all this stuff. Nemo. That's a whale. No. But I didn't hear a word she said for several seconds because I'm all focused on, do I need to know what the LZ means? No, I didn't need to know. And I didn't need to know all about this degradation either. The only thing I needed to know is that TNF alpha, when it engages the receptor, sets in motion a process that leads to gene regulation. That's what I needed to know. And ultimately, when she repeated the talk, that's the slide she developed. Because you couldn't possibly explain all of this in a 20-minute talk and give results. And if you just gloss over it, you do what happened to me, and I just focused on things she didn't want me to pay attention to. Okay. Roadmap slides. They lead the audience through the talk. I used the message slide. A roadmap is an outline, whatever you want to call it. It leads the audience. It tells them where you're going, where you are in the talk, and some key points. It's the storyline. It's the chapters in your story book. Okay, here's a slide that I used. This was at a Keystone meeting. They wanted me several years ago to talk about the historical background about ApoE and then about its role in neurodegenerative disorder. So I used this. I said this is what we are going to cover. I used it several times. It told the audience where we were going, what I was actually going to describe, and I used it, as I said, several times. So after I had done the first 2 points, then I focused on Alzheimer's disease. It was a transition slide. It was a roadmap slide, and it actually had elements of my message. And when I got to a particular point, I highlighted it in yellow so they knew where I was. I used that same sort of slide in the demonstration that I told you about, where I highlighted different points. I led you through the outline, the message, the roadmap, if you will. In my case, it was, in fact, the message as well. Method slides, very important. Consider the best way to describe a method, and you have to do it quickly. You don't have time for a lot of details in most cases. Now, if it's primarily a methodologic talk, of course, that's not what I'm referring to. I'm talking about general talks to more general audiences. You want to let them know what you did, how you did it, but you don't want a lot of the details. You want to have time to focus on your experiments, on the results, and on your data. So here's a method slide. It's okay. The method was looking at the remodeling of dimerystyl phosphatidylcholine by ApoE. So to study this, you produced these phospholipid vesicles. You trapped a fluorescent dye in the vesicle. You added ApoE, and you measured the dye that was released. Okay, that's fine. That's not bad, but I consider this better. It's exactly the same thing. You form the phospholipid vesicles. You trap the fluorescent dye in the vesicle, and when you add ApoE, you remodel the vesicle, and you release the fluorescent dye. Maybe you agree, maybe you don't. It's the same thing, but I find, again, a model, a picture is worth 1,000 words. And I found this. In fact, the class helped to design this slide. The student, he used this slide. We said it was okay, but couldn't you make it a little bit more interesting? Couldn't you give me a few more helps as to what you were actually doing? And so we suggested this. Now, transition slides. We've already talked about this, that you use them to change the topic, to introduce a new topic. You can do various types of transition slides. Question slides work very well. For example, in this particular talk, there were two major questions that the individual wanted to present. One was, does ApoE interact with free fatty acids, and does it alter the concentration of free fatty acids? So he, in this case, used this slide twice, highlighting where he was in the talk, used as a transition slide. A few years ago, I gave a talk in which I related ApoE4 to traumatic brain injury and Alzheimer's disease. This was a transition slide. I didn't really say anything at all about this because I had just spent the beginning of the talk talking about this, but it was a transition slide because I then said, what I would like to show you now is that ApoE4 actually represents a new therapeutic target. It's a visual way to say we're changing topics. I'm now going to tell you about ApoE4 as a new therapeutic target. You can use questions. I've already said that. I don't think I have to say any more about that. What's the best way to present your data? Always consider the most visual way, the way that the audience can capture the essence of your data the quickest. Tables, of course, bar graphs, figures, that's the ways that we display our data. So here's a table. Again, I apologize for all the ApoE, but it's what I have easiest to show you. And so here, actually, we're looking at ApoE binding to fat-rich lipid particles. And what this table shows is that ApoE4 binds to a much greater extent than does ApoE3. But when we treat ApoE4 with a structure corrector molecule, it now behaves, the E4 behaves like the ApoE3. Well, that's the facts, that's the data. But someone suggested that it would be more visual if I presented the data this way. ApoE4 binds to a greater extent to these fat-rich emulsions than does ApoE3. But when I treat ApoE4 with a small molecule structure corrector, it now behaves like ApoE3. You got the same data, and maybe you agree, maybe you don't, that this is a better way to present the data. It's more visual. That's up to you to decide. So now, let's have some more general considerations or more general recommendations about specifics in slides. So let me start with general recommendations. Typically, on a word slide, no more than 8 to 12 lines of text, and usually no more than 8 to 10 words on a line. That will allow you to keep the font size large. That's your goal. You can use that slide then in any conference room or auditorium setting if you follow some of these general recommendations. This actually is 7 lines, and this, I believe, is 8 words. Use short phrases. You don't read the slide. You use the slide as a guide for your audience. You use colors to focus attention on key points. You just don't put color everywhere for the heck of it, because what do you see when you see color on a slide? Your eyes go right to that. You focus. This must be important because he or she put it in blue. Well, make sure that when you use color, it's something that a point that you're trying to make sure that the audience gets. And use space appropriately. Don't have a bunch of just blank space on a slide. Fill it up, make the font size larger. I'll say a lot more about that in just a few minutes. So let's talk about slide titles. We've already talked about the two different types of titles. Now let's say a little bit more detail about the titles. Every slide should have a title. That's just best practices observed by lots of people. Every slide should have a title. It's very helpful, as we've already said, but it's also the first thing that you look at, so that make the title important. Capitalization. Again, this is best practices. You don't have to do this. It's best practices. You decide. But you decide and then be consistent. There's nothing worse than to have hodgepodge lodge types of slides in which you follow all sorts of different guidelines. Every slide in your deck should look like it's from you, and it shows that you've paid attention. So what I believe is best practice, capitalize the initial letters of all words, just the first letter, just like I've done it here, except articles, conjunctions, and prepositions. Best practices, not law, best practices. Always capitalize the first and the last word. Obviously, the last word can't be an article, a conjunction, or a preposition, so the last word by default would be capitalized. I probably should take that off. It always takes me several seconds to explain why I put it on the slide, and it's obvious once you think about it. And do not use all capital letters. They're hard to read. And don't use funny fonts with squiggles and different shapes of letters. Make it simple. KISS. Keep it simple. Stupid. Okay. I didn't say that. Somebody else said that. All right. Point size of titles, 32 to 36. That's a guideline. Maximum 3 lines. It's better, the shorter the title, to be meaningful, the better, because the audience wants to read. Fonts. I like Helvetica, and I bold all text. It's easiest to read. Best practices. Font sizes, never less than 20. I actually say never less than 24. Use 36, 32, 28, 24 if you need to. Don't have a whole bunch of different font sizes on a slide. Be consistent and use italics and color for emphasis, but not a lot of fancy typed. So let's talk about color. We've already said color on a slide immediately focuses your eyes. It immediately focuses your attention. It's for emphasis. And as I said, it's for focus. Now the background of the slides. I obviously like dark. I obviously like black because every slide I've shown you is this way. A lot of people, a lot of PIs like a white background. That's your choice. I like the dark black or blue. For me, it's easier to see. It's not so bright in most lecture rooms, and you have nice contrasting white text. As we've said, use color to focus the attention of your audience. The best colors are yellow, blue, green, and red, but remember, don't use red and green on the same slide because of colorblind men, which is not uncommon. Limit the colors. Don't just, as I said, have a whole smear of different colors. Doesn't do you any good, and it actually detracts from your data. Now, the Gladstone graphics department actually created this title for me to irritate me, to actually emphasize the use of multiple colors, which I, of course, don't agree with. But they did this just for the heck of it, and that's a color palette. Consistency among your slides. ApoE4 is always yellow. ApoE3 is always blue. It's simple. Every slide should be consistent with the color scheme that you use. If you color a certain protein or a certain process, color it the same on every slide. It's just another crutch. It helps. And I won't go through the rest of the data. You choose the colors you like. I like the light colors. Never use dark red. I don't really like purple and violet. Sometimes, depending upon how much light there is in the room, especially violet, it's hard to read what's under the violet color. As I said, I like yellows, light blues, light reds, even pink, something that does not obscure the words that you're highlighting. The same is true for PowerPoint. Choose the colors, choose them carefully. Now let's talk about formatting of slides. And I will just show you now some different slides. You can look at the information on this slide that you have in your deck. But what I'd like to do as I end the lecture is to show you examples of good slides and bad slides. For example, for me, this is a bad slide. The font size is just too small. There's just so much wasted material. Why not increase the font size? The gentleman on the back row's got a hard time reading this. That gentleman may or may not know whether these are plus signs or not. Plus and minus are very difficult to read. And if you're working with transgenic mice, it's an entirely different world if it's a plus wild type or a minus knockout. And often you'll see slides in which they indicate a wild type or a knockout mouse with a minus, and you can't tell whether it's a minus or whether it's a plus. So pay a special attention with pluses and minuses, but also pay attention to the font size. This is such a much better slide. You filled the space. It's the same data as on the last slide, but now anybody can read this because those font sizes are in that range that I talked about, 24 to 36. You can read this slide in any room, in any lecture hall. It's so much better than that slide. It's the same thing, but it's just much better. Now, this is a slide that was, again, used in the course. It has to do, again, with ApoE. And this was her outline slide with the different components. It's very difficult to follow. You're not exactly sure when things end and when things begin. And so we suggested to her that she do something more like this. In the first place, fill the slide with the text. And now use bullets for a subpoint and a sub-subpoint, use a dash. Now it's just a much easier slide to read. It's much more interpretable as to what she really had intended for that slide to be. Here again, another slide, not a horrible slide, but it can be much better because look at all this wasted space. Why not just fill it with the information so you don't have to worry? The gentleman on the back row can actually not read, I believe, this ordinate. It's just the font size is too small. So increase the size. Make it readable. Make it easy for your audience. And finally then, about animations. You can use animations in various ways. I don't like really, I've already explained to you I'm pretty conservative, so I don't like things flying in and flying out and that sort of thing. But I do like step slides. I didn't actually happen to use any this time, I don't think. Maybe I did in the first lecture, where you do point 1, then you bring in point 1 and point 2 and talk about point 2, and then bring in 1, 2, and 3 and talk about 3. I believe I did show one such slide as that. Another use of animation are methods, and this worked very well. The remodeling of a phospholipid again by this protein. It has a big impact, and you can mutate the protein. It changes the way it behaves, and you end up with something like that. So stepping that slide worked very well. Was somewhat too complex if you just showed the whole thing at once. Maybe not, maybe yes, but stepping from my point of view, made it a little bit better.